Welcome everyone. Thank you for joining us today. It's going to be a marathon of 21 presentations, so quite a feat to coordinate all that. But we are thrilled to have such outstanding presenters, and I hope you will all enjoy and learn from this today. So the Medicine and Public Health panel speaker today begins with Dr. Mioara Larion, and her presentation today will be about breaking barriers in science and life. She's trained as a biochemist, and she completed her Bachelor's of Science from Alexandru Ioan Cuza University in Romania, and continued to study the same topics for her master's PhD at Florida State University. She did her graduate school in the lab of Dr. Brian Miller, involved in biophysics and enzymology, enzymes. And during that time, she developed a passion for learning about enzymes functions and how the dysfunction can cause or exacerbate certain diseases. After completing her PhD, she continued her postdoctoral training in nuclear magnetic resonance with Dr. Bachweiler at Florida State University and Ohio State University. And after that, she joined the Neuro-Oncology branch as an investigator, and was introduced in the field of oncology at the Cancer Metabolism Research Program, one of the labs at the NIH, the National Institute of Health. During her tenure at NIH, she had multiple publications in Nature Methods, Nature Communications, Transient Cancer, and others. She's involved in several committees, and she's the associate editor of the Journal of Cancer Metabolism, one of the Spring in Nature journals, so highly prestigious, and lots of other positions and involvements that it's beyond my my level of comprehension to enlist here. I would like to give her the floor for her presentation.
Good morning, everyone, and thank you so much, Theo, for this kind and nice introduction, and for your invitation to be part of this group. I'm very excited today to present to this group, and I'm going to share my screen. I hope everyone can see it and can hear me okay. So today I thought about going over my career path, as Theo suggested, and describe how I end up doing what I'm doing in the National Institutes of Health. As Theo mentioned, my lab is located in Bethesda campus, which is in the National Institute of Health, part of the National Cancer Institute. This is an intramural program funded by the NIH, by the United States government. This in the middle picture, I show Building 10, which is the national largest hospital, and where many of the first in-human clinical trials are happening. And in the right, I show a picture of Building 37, where my lab is located. And I wanted to advertise that there's a large intramural training and education program at the NIH, and a lot of postdoctoral fellows as well as PIs are international, and they come from all over the world. I was born in a very small village called Solești in Vaslui, a second of 7 kids. And my parents recognized my passion for science very early on, and so they sent me to a better school at the 5th grade level in Vaslui. After that, I went to the high school, theoretical high school, called Liceu Mihail Cogălnicianu. This is where I met my mentor, a high school professor, Adriana Ciupercescu, who instilled my passion for organic chemistry through chemistry Olympiads at the time. Then I graduated from high school and attended Alexandru Ioan Cuza Iași in chemistry and biochemistry. And at the end of, I want to mention, I'm the first generation college graduate. At the end of my college degree, I applied via an opportunity and under the mentorship of Dr. Lenuța Cireș to the PhD program at Florida State University. So immediately after graduating from my undergrad, I attended FSU. Here, it's where my scientific career started. I started with spectroscopy in the lab of Dr. Peter Fire, and our project was to test the first W-band instrument that came from Bruker in one of the national labs, the National High Field Magnetic Lab at Tallahassee, Florida. And this is one of the labs together with Los Alamos and Argonne. And at the end of my master's, where I was testing this instrument to see the sensitivity and the dynamics of this new instrument to sense motion of proteins, after I defended my master's in biophysics, I joined the group of Dr. Brian Miller, who's an enzymologist, and I rediscovered my passion for organic chemistry, but done very efficiently by this catalyst called enzymes, which are very important for our health, in fact, and they are instrumental for metabolizing all the food that we are eating. In this lab, my project was to find direct evidence and to understand why human glucocorticoid glucokinase functions different than any other enzymes. And the role of this glucokinase is basically to sense a glucose concentration via a unique sigmoidal dependence of the reaction as a substrate concentration shown here lower versus normal Michaelis-Menten hyperbolic type of enzymes. And there was a postulate from Jean-Pierre Chargeot, which I displayed here from Institut Pasteur, who in 1950s suggested that different conformations of enzymes could account for this type of behavior in monomeric enzymes, because allosteric is normally associated with protein binding, for example, oxygen binding to hemoglobin. In any case, my goal was to get this direct evidence, and in order to do that, I studied many years, applied many techniques. And one technique that was particularly interesting to me at the time, and which I mastered in my postdoctoral fellowship, was nuclear magnetic resonance, because this allowed to look at protein structures in solution. So we could detect conformation differences in solution, so it wasn't artificial. Here I had the opportunity to work with Raphael Bruchweiler, who is a world expert in NMR, and I learned all about NMR, and I applied it to my problem. And we found the direct evidence for this unique sigmoidal kinetics. We characterized the conformation of the enzyme in solution, and I obtained this again, another spectroscopic technique under in my toolbox to say. Towards the end of my postdoctoral fellowship, I was looking for interesting enzymes, and in New England Journals of Medicine, there was a report that mutations in a metabolic enzyme called isocitrate dehydrogenase 1 are linked with glioma. This is a study, the first identification via a genome-wide screen done also by with the co-author, which is a Romanian co-author, Victor Velculescu, and they show that majority of these gliomas have a mutation in IDH1 and IDH2 in the active site of the enzyme, and this mutation is associated with better survival in patients. So I found my project to start my own lab. I was very excited about this new direction that my postdoctoral advisor or PhD advisor were not interested in, and I applied to many universities in 2015, and NIH selected me from 100 applicants to start a group that focuses particularly on studying metabolism in this type of gliomas. And for that, I wanted to save some time and maybe present one of the short videos that my branch made very recently about my group. And their energy to function, and they do that using a set of biochemical reactions to fulfill those needs. The sum of all these biochemical reactions forms metabolism. However, cancer cells have no inhibition, they evade cell death. For that, they require certain metabolic reactions to grow and divide. My name is Mioara Larion. I'm a tenure-track investigator leading the cancer metabolism program in the Neuro-Oncology branch of the National Cancer Institute. By understanding metabolism, we will be able to develop better, less toxic anti-cancer therapeutics. More than 80% of gliomas, which are a common brain tumor, have a mutation in a metabolic enzyme called isocitrate dehydrogenase 1, or IDH1. My lab is very interested in understanding this because it's one of the very few examples of a tumor driven by a mutation in a metabolic gene. We think this offers opportunity to identify metabolic drug targets. Cancer cells require lipids or fats for their cellular components, for their energy demands, and for their signaling needs. IDH1 enzyme is very important for synthesis of these fats or lipids. We think that brain tumors with an IDH mutation are particularly going to be susceptible to lipid-based metabolic targeting. We just need to understand the difference between IDH mutated lipid metabolism and normal cell lipid metabolism in order to identify these pathways. My lab employs a collection of methodologies from standard cancer biology techniques such as Western blots and drug viability studies to more complex techniques to study such as mass spectrometry and spectroscopy. Very unique to our group is the ability to image and quantify lipids using Raman spectroscopy in organelles of live cells. This is something that sets us aside and that enabled us to discover lipid pathways. Besides this unique feature of our group, we also employ more classical ways to study metabolism, such as carbon-13 tracing analysis, in which we are able to look at the fate of a nutrient in cancer cells and normal cells. In 2021, we discovered that IDH1 mutated gliomas have a unique ability to produce more monounsaturated fatty acids, which are a particular type of lipids with a unique chemical structure and a one double bond. We think this is one example, one pathway that we can target metabolically. We also found that in IDH mutated glioma, certain lipids called sphingolipids are in high abundance. And currently, we are in the way to identify targets of the pathway specifically for IDH mutated glioma. My favorite part about working in the Neuro-Oncology branch is to see how our discoveries in the lab are helping patients prolong their life and improve their outcome. By understanding what lipids are good for us and what lipids are not so good for us, we could have a better lifestyle, a healthier lifestyle, and prevent the formation of diseases. Okay, this helps me to focus on one technique and describe in more detail one of the technologies that we're trying to develop and how we're looking at metabolism in a very unique way. So in about 2017, Thermo Fisher came up with a new machine, a microscope that they were bringing to the market, and they were looking for someone at the NIH who could test this in biological samples. And Adrian Lita, another Romanian, was one of the few experts who knew how to Raman spectroscopy is the technique itself. He used that in his graduate studies for inorganic chemistry. So he started testing this machine, and we developed a lot of methods based on Raman spectroscopy, but most important, and I will go over some of the methods that we are focusing on. One advantage of this technology is that we could look at metabolism in a very unique way. And I'm telling you a little bit more about this. Physically is a surface scanning technique. It scans the sample, the tissue, the cells in their microenvironment, and it produces this unique chemical vibrations. And they represent, as shown here in this dynamic, they have a peak in the spectrum, and it creates molecular fingerprints of the area. But the important part for us is that the intensity of the peak is proportional with the concentration of the material, so it allows us to quantify composition of cells or tissues. And so you could think about this technique as transforming a black and white picture into a more colored picture. And here I have a video where from the Thermo Fisher machine showing how the microscope works. We're scanning pixel by pixel the surfaces and creating for each pixel a unique spectrum. And this spectrum contains chemical information, therefore creating these molecular fingerprints. Now, we're applying this to because it's a microscope and a confocal microscope, it has a resolution of 500 nanometers, so we can look at inside of the cell. So instead of just looking at the cell, we can scan inside of the cell. And for that, we developed methodologies that we adapted this machine with the help of a fluorescent filter that we can identify these organelles and measure their chemical composition, organelle by organelle. And I think I want to highlight the contribution of Dr. George Palade, who was born in Iași and got Nobel Prize in Medicine in 1974 for his efforts to understand organelles. I think you'll be very excited to see how we can study organelles in their microenvironment without the need to extract them. And so we are applying this to look at different tumors and the tumor microenvironment and dissect the difference between normal cells, tumor cells, immune cells, based on their chemical composition. And here's an example how inside of a cell you could map DNA, lipids, and proteins, or you could look at different cells as a function of different drugs. We could also look at the interaction between cells that are or connections called tumor microtubes. And of course, we can map different biomarkers or drugs as long as they have any chemical composition. We can apply to tissue. And here I want to highlight a project that I started a few years ago in collaboration with Dr. Joon Petre at the University of Turku in Finland, who is a computer scientist and a mathematician, and helped us design machine learning algorithm that makes our life easier to classify this tumor based on their chemical composition. So our project started a few years ago, and we also added other group in Romania at the University of Bucharest at the Faculty of Mathematics and Computer Science, led by Dr. Andrei Păun. And basically, they're helping us to identify via machine learning and the differences between different tumor types and classifying them and figuring out what chemical composition is particularly unique for one type of glioma. And I think this it's already having an impact on surgery because there is a Raman spectroscopy-based instrument used in the operating room by Daniel Orange and others who are using it to guide tumor biopsy to see in real time visualization of the margin. And here is an example where you can see in the bright field, there's no difference in the tumor versus the normal tissue. But if you apply this microscope with a fiber optic, you could see the difference between tumor and normal, and you could see the tumor infiltrating cells that are very difficult to observe by neurosurgeons with the naked eye. And this is my current lab, and I leave you here with the quote that the only way to discover the limits of the possible is to go beyond them into the impossible, by Sir Arthur Clarke. And I think this applies also to Theo's efforts. So thank you so much for your invitation and for listening.
Thank you. Thank you. That is very sweet of you. And